Hello, friends. Today we are going to study about internal structure of monocot stem. Internal structure of monocot stem. The typical example which we are going to visualize is maize plant, maca plant in Marathi. All the monocots they show same internal organization of the tissues, internal framework of the tissues, but we are going to visualize maize as the typical example. Last time we have studied the internal structure of dicot stem. Today about the monocot stem. The monocot stems, for example, maize, jawar, rice, wheat, sugar cane, then coconut, areca nut, betel nut. These plants are the monocot plants. They show no secondary growth. But we have to visualize how the stem is there internally. What internal primary organization the stem it shows that we have to study. What type of vascular bundles are there, how they are organized, and what are the different tissues, different regions internally composing the stem, and what functions they perform. Now, for example, over here, this is the transverse section of the young monocot stem maize. Now, this internal structure of the maize stem, monocot stem, is totally different from the dicot stem. It shows outermost covering as epidermis. Below the epidermis, there is a hypodermis. Hypo means below. Below the dermis, below the epidermis, there is hypodermis. Now, over here, there are 2 to 3 layers of the cells composing the hypodermis. This hypodermis is a mechanical tissue in the monocot stem. Over here, this hypodermis is composed of the sclerenchyma type of tissue. Hypodermis is made up of sclerenchyma type of tissue. We have studied the hypodermis of dicot stem. There, the hypodermis was made from cholenchyma type of tissue. Over here, there is a sclerenchyma, hexagonal, pentagonal, just like the honeybee copse. The structure of the sclerenchyma cells, they show their structure. And this particular hypodermis present below the epidermis is the mechanical tissue, giving the mechanical strength, rigidity, strongness, toughness to the monocot stem. So this is about the hypodermis. Now below the hypodermis, up to the center, there is a ground tissue. This is a ground tissue. This is a ground tissue. All this is the ground tissue. And scattered in the ground tissue, there are the vascular bundles. Vascular bundles are scattered in the ground tissue. They are randomly scattered in the ground tissue. Vascular bundles are randomly scattered in the ground tissue. Now we have seen the arrangement of the vascular bundles in the dicot stem. There in the dicot stem, the vascular bundles were limited in number and they were organized in a concentric circle. They were arranged in a ring manner. Over here, the vascular bundles are irregularly scattered. Suppose one is over here, second is over here, third is over here. They are randomly scattered. Randomly means they are irregularly distributed in the ground tissue. So this is the feature of the monocot stem, that the vascular bundles are randomly scattered. And beside this, the vascular bundle they shows one more feature. The vascular bundles toward the peripheral region, that is toward the outer region. For example, this is the outer region. This is the outer peripheral part of the stem, where the vascular bundles are smaller in the size and they are more crowded towards the periphery, towards the outer part of the circular stem. And whereas there in the central part over here, the vascular bundles are larger in the size. They are slightly larger in the size and they are little in the number. They are little in the number, or we can say the total number of vascular bundles in the core part, central part of the stem, they are very scanty, little in number. So this is about the ground diagrammatic section, what different layers it shows. It shows epidermis, hypodermis, there is ground tissue, and scattered in the ground tissue there are the vascular bundles. Now of this total ground plan diagrammatic section, we have to see the internal structure of the different cells, tissues composing the different layers, and we have to also visualize the structure of the vascular bundles and the different functions the vascular bundles and different regions of the stem internally they perform. So for this purpose, we can visualize the sector of the stem. For example, this is the sector of the stem. We have to observe what structures from the epidermis up to the core part it shows, and how is the structure of the vascular bundles. So this we have to visualize only the sector. We have to visualize only the sector. This much part we have to observe in detail. So this is about the ground plan. Now over here, this is the sector of the stem. This is the sector of the stem. This is the sector of the total round section showing the regions from epidermis, hypodermis, and vascular bundles. So all the structures are seen from periphery up to the center, up to the core part of the stem. So this is enough to describe the structure of the total ground plan, or total complete section of the stem can be explained by visualizing this much sector part of the stem. Now we have to prepare this figure for examination. We have to prepare this figure for examination. It is not necessary that you should prepare. This is the ground plan. This is just for understanding. This is just for understanding. This figure is important for examination angle. For examination angle. Now, in addition to this figure, over here, this is the very beautiful figure labeled, and in addition to the labels, the different functions and composition of the different layers, what tissues they compose the particular region, and what function the particular region will perform. These things are written over here. You can write only the labels, the functions and the tissues, type of tissues. It is not necessary to write, but you can mention the type of tissues composing the particular layer, and the functions can be written. So let us describe the internal structure of the stem. What different layers it shows. The outermost layer is epidermis. This is the epidermis. Epi means outer, and dermis means a layer or a skin. Outermost skin, just like our body have the skin, which is protective in function. Here, there in the monocot stem also there is the epidermis. Epidermis is the skin of the stem performing the protective function. Now, to perform this protective function, the epidermal cells are compactly placed. They are barrel-shaped or cylindrical in the outline, and they are compactly placed without intercellular spaces. And to perform this function of protection nicely, this particular epidermis is deposited with the extra cuticle. This is a coating. Epidermis is deposited with the extra coating on it. Now, this extra coating or extra protective deposition on the epidermis is called cuticle, which helps the epidermis to perform the function of protection nicely. So this is all about the epidermis. Outermost protective skin of the stem is called epidermis. Now, below this epidermis, there is a hypodermis. Hypo means below, lower. Dermis means a layer. Below the epidermis, there is a lower layer, lower skin. We call it as hypodermis. This hypodermis is made up of sclerenchyma type of tissue. Sclerenchyma, this is a sclerenchyma type of tissue. Just like hexagonal, pentagonal in the outline, it have the hexagonal or pentagonal outline, just like the benzene ring which we sketch by this fashion. So this is a sclerenchymatous hypodermis. We have sketched these sclerenchyma there in the dicot stem in the pericycle. So this sclerenchyma were there in the pericycle in the dicot stem. Here they are there in the hypodermis. Here they are there in the hypodermis. Now you can remember in the hypodermis of the dicot there was cholenchyma type of other tissue. Over here the hypodermis in the monocot stem is made up of the sclerenchyma type of tissue which are hexagonal or pentagonal in the outline. Now this hypodermal sclerenchyma, this is a mechanical tissue, very strong, rigid and mechanically strengthful tissue giving the mechanical strength to the stem. So whatever may be the height which the monocot stem can achieve, this height should be sustained. For example, bamboos grows about 100 feet, 80 feet, 50 feet in the height. Sugarcane grows about 15, 20, 22 feet in the height. Maize 12, 15 feet height. So whatever may be the height which the monocot stem can attain, so this height should be supported, sustained, and for this purpose, this mechanical hypodermis, mechanical tissue, hypodermis, sclerenchyma tissue composing the hypodermis is very important to give the strength to the stem. So this is all about the composition and function of the hypodermis. Now below the hypodermis there is a ground tissue. Below the hypodermis there is a ground tissue. This is a ground tissue over here, ground tissue. So below the hypodermis up to the central part, core part, center of the stem, all round there is hypodermis. There is a ground tissue. And this ground tissue is composed of the parenchyma type of tissue, which are loosely placed, thin-walled, with numerous intercellular spaces. So between the cells there are intercellular spaces. Empty spaces are there, storing water and other substances. Intercellular spaces are there. And as such, this total ground tissue is the storage tissue, storing water and starch and other reserve food substances in different monocots. This is a storage tissue. This is a storage tissue which is very massive and soft, delicate. So this is about ground tissue. Now scattered in the ground tissue there are the vascular bundles. In the ground tissue, vascular bundles are randomly scattered. For example, these are the vascular bundles. This is one vascular bundle, this is another vascular bundle, this is third vascular bundle. Such numerous vascular bundles are scattered. Randomly scattered, irregularly scattered in the ground tissue. So there is no definite fashion of distribution of vascular bundles like the dicot stem. In the dicot stem, the vascular bundles were arranged in a concentric manner, in a ring manner, in a circle. Over here, the vascular bundles are irregularly distributed in the ground tissue, irregularly scattered in the ground tissue. So this is one more difference from the dicot stem, how the vascular bundles are organized in the monocot stem and how they were there in the dicot stem. Now in addition, we have to describe the structure of the vascular bundles. The vascular bundle have the two principal components. One component is called xylem, another component is the phloem. Now this xylem and phloem, these are joined together in one bundle. So this is called as conjoined. When xylem and phloem are jointed together in one bundle, the bundle is called conjoint. Xylem phloem jointed together forming a one bundle, we call the vascular bundle as conjoint. Now depending upon how they are located, the vascular bundles are called collateral. Collateral means, suppose lower is the xylem, above it is the phloem, one above the other. One above the other. Suppose this is xylem, above it the phloem is placed. This is xylem, above it the phloem is placed. Blue color is the phloem. So this is such a arrangement of the xylem and phloem, one above the other, just like the steps of the building. We call such vascular bundles as collateral vascular components of the vascular bundle, xylem and phloem. They are collaterally placed one above the other. Now, beside this, these vascular bundles, they shows no cambium in it. We have seen the dicot vascular bundles, and in between xylem and phloem there was the cambium. Here, these vascular bundles shows no cambium, and therefore, when the cambium is absent in the vascular bundles are called closed type of vascular bundles. The vascular bundles are called closed type of the vascular bundles. Now, as the cambium is absent, vascular bundles are called closed. The monocot stems, they shows no process of secondary growth. Secondary growth means increase in the thickness of the stem. Increase in the thickness of the stem. Now we have seen the maize stem, sugarcane stem, supari stem, coconut stem, bajra stem, wheat stem. These stems, they do not increases in the thickness, increases in the girth, because there is no secondary growth. The monocot only increases in the height. There is no increase in the lateral increase. There is no increase in the circumference girth of the stem. Do not increases, as there is no process of secondary growth. The vascular bundles are closed. There is no cambium in the vascular bundle. The secondary growth is absent. So these are the three words describing how the monocot vascular bundles they are. Monocot vascular bundles are conjoint, collateral, and they are of closed type. So these are the three features of the vascular bundles of the monocot stem. Now, in addition to these, the monocot vascular bundles are also characterized by the bundle sheath. Bundle sheath, there is a bundle sheath over here. Sheath means a kavach, a protective covering around the xylem and phloem in the vascular bundle. Now, this bundle sheath totally covers the vascular bundle. So, all this is the bundle sheath. This is sclerenchymatous bundle sheath. This is sclerenchymatous bundle sheath. Bundle sheath means a protective kavach, coating, sheath, cover around the xylem and phloem. This is called as bundle sheath. This is called as the sheath. Sheath means a kavach, and it is made up of harder sclerenchymatous tissue. It is made up of harder sclerenchymatous tissue. So, presence of bundle sheath is a feature of monocot vascular bundle. This bundle sheath is absent there in the dicot stem. Dicot stem shows vascular bundles where the bundle sheath is absent. Here, the bundle sheath is there in the monocot stems. Now, in addition to this, if we observe the elements of the vascular bundle, xylem and phloem, suppose if we observe the xylem, this xylem is V-shaped. This xylem is V-like, capital V-like or capital Y-like, capital Y-like or capital V-like. The xylem and phloem, suppose this is one xylem, this is another xylem, this is smaller xylem. The metaxylem and protoxylem, we respectively call them as the larger elements are called metaxylem, the smaller elements are called protoxylem. They are organized in a capital V manner. And above the arms of the V over here and here, there is a phloem over here. This is a blue color is the phloem. Suppose this is the V-shaped xylem. This is the phloem. Blue color is the phloem. This is the V-shaped xylem. This is the blue color is the phloem. So this is how the xylem and phloem is organized. The xylem is made up of metaxylem, protoxylem, tracheids, and tracheids means the conducting tubes, vessels, conducting tubes for the conduction of the water. These are the capillaries conducting the water in the monocot stem. So this is the xylem tissue, water conducting capillaries, water conducting tubes present in the vascular bundles. Vascular bundles are the conducting strands. Now, in addition to the xylem, this is the phloem. This is the phloem. Above the arms of the xylem, this is xylem, this is xylem, this is xylem. The phloem is located over here. So, this is about the location of the phloem. The phloem is located above the xylem. This phloem is also made up of protophloem, metaphloem. These are the types of the phloem which are derived from the primary meristem. The function of the phloem is translocation of the food. The food, glucose prepared by the photosynthetic green leaves, this should be transported to the rest of the stem, to the roots and rest of the organs of the stem. The phloem serves this function of transportation of the food. The food is transported to the different organs. We can call transportation or translocation from the leaves to the different organs. So this is the function which the phloem performs. Now, in addition to the xylem and phloem, if you observe the vascular bundles, this shows this cavity over here. This is cavity over here. This is cavity over here. This is called as water cavity, or it is also called as lysigenous cavity. Lysis means a breakdown. Lysis means a breakdown. So what is broken over here? The smaller protoxylem points, the smaller elements of the xylem, we call them as protoxylem. So this is the protoxylem over here. These are the protoxylem elements over here. When the protoxylem elements they get broken, so there is empty space, cavity, duct is formed. This empty cavity thus formed stores the water. So therefore it is called as water cavity. And as it is formed by the breaking lysis of the protoxylem elements, we can call this as lysigenous cavity. This is present toward the lower side of the vascular bundles. So this is how the vascular bundles of monocot, they are very characteristic. They have the bundle sheath, they have the V-shaped xylem, they show the water cavity over here, they shows a phloem above the arms of the xylem, and they shows no cambium. The vascular bundles are called closed. Now, beside this, these vascular bundles of monocot, they shows a oval shape. We have seen the dicot vascular bundles. Dicot vascular bundles are top-like. They are broader toward the apex and tapered toward the base, just like the top. We called it as a wedge. Vascular bundles are wedge-shaped. Broader at the apex, tapered toward the base. But over here, these vascular bundles are oval in the outline. In the monocot stem, they are oval. Vascular bundles are oval, whereas In the dicot stem, they are wedge-shaped. Wedge means just like this. This is a wedge-like shape. Whereas in the monocot stem, they are oval in the outline. Oval in the outline. So this is the difference from dicot vascular bundle shape and monocot vascular bundle shape. Now beside this, here these vascular bundles are randomly scattered, one over here, another over here, third over here, and this is how they are randomly, irregularly distributed in the ground tissue. They are irregularly distributed in this ground tissue. So this is one more difference between the arrangement of the vascular bundles of monocot stem and the dicot stem. So this is all about the structure of monocot stem, right from epidermis, below there is hypodermis. The hypodermis is made up of sclerenchyma type of tissue, and when the stem is very green and tender, young, green colored, this hypodermis may show certain photosynthetic cells in certain patches. This we call this tissue as chlorenchyma type of tissue. We call this as the chlorenchyma type of tissue. If the stem is matured, this hypodermis do not show chlorenchyma. You can sketch chlorenchyma over here just like this figure, or you may not sketch the chlorenchyma. It is not necessary that the chlorenchyma should be sketched, must be sketched. It can be avoided. You can sketch only this sclerenchyma in the region of the hypodermis. It is not necessary to sketch the chlorenchyma, but if it is a very young stem, green in color, there it shows the chlorophyll containing photosynthetic cells, tissues in the form of patches of chlorenchyma there in the hypodermal part. This is hypodermis, and in this hypodermis, the chlorenchyma may be present when the stem is very young and green in color. Young and green in color. So hypodermis is there. Below the hypodermis, there is a ground tissue, and randomly scattered in the ground tissue, there are oval vascular bundles. There are oval vascular bundles. The vascular bundles are conjoined, collateral, and are of closed type, and each vascular bundle is provided with a bundle sheath. Protective coating is there, protective coverage is there, protective sheath is there. We call it as bundle sheath, and each vascular bundle shows a water cavity towards the lower side. So this is about the internal organization of the monocot stem. Now if we compare this structure with the dicot stem, there in the dicot, there was the epidermis, hypodermis. Below the hypodermis, there was cortex. Below the cortex, there was the endodermis. Below the endodermis, there was pericycle. Below the pericycle, there was the ring of vascular bundle. And in the center, there was the pith. So different layers were there in the dicot stem. Epidermis, hypodermis, cortex, then endodermis, pericycle, ring of vascular bundles, and the center part, pith. So such differentiation of the different layers there in the monocot stem, it is not seen. Monocot stem just shows epidermis, this hypodermis, the ground tissue, and scattered in the ground tissue are the vascular bundles. Scattered in the ground tissue are the vascular bundles. So we have to prepare this figure, sector of the figure. All diagrammatic ground plan figure should not be prepared. This is important in the angle of examination. So this should be sketched, and it should be beautifully sketched. This should be shown darker. Vascular bundle should be shown darker, and ground tissue should be sketched light in color. And such a majestic, beautiful figure attract the attention of the examiner. So one can sketch the beautiful figures like this. Both the dicot stem and monocot stem, I have chosen a very beautiful and very nice figures for a sketching purpose. So this is all about the internal organization of the monocot stem. Typical example is maize. So we have to prepare accordingly. And I have also given you the visual content of this monocot internal structure. You can sketch the figure. The description should be written from the visual content, and still if you have any doubts, you can ask me the questions on the group. So with this I conclude over here. I say Dhanyavad.